All right, folks. So to understand the paper on Friday, there wasn't a very good iBiology video or paper, so I decided to make my own. So this is my attempt at a very budget version of an iBiology video. And the paper on Friday looks at oocyte development in a multicellular organism. The oocyte development is obviously very important because that's what gives rise to the next generation of animal. And so there's lots of positive selection pressure to have this go well. The model system that we're using to study oocyte development is Drosophila melanogaster, the fruit fly. And the reason is because they have this really neat feature where if you take an adult female fruit fly and take out its ovaries, the ovaries are comprised of these string-like structures, which are the ovariole. And these oocytes, or the egg chambers, bud off from the stem cell population of this tissue and then begin to develop as it moves along. So this is a later stage egg chamber, this is an earlier stage egg chamber, and what it gives you is lots of snapshots of development over time. So we could look at this another way. So again, illustrating that the earliest egg chambers to bud off from the stem cell population is obviously the earliest stage. And as you move out along this string of pearls, you start to get to late stage egg chamber development. And what we're looking at is the development of this oocyte. So at this stage, it's mostly just comprised of nurse cells, and nurse cells function to support the development of the oocyte. So over time, through the stages, nurse cells squeeze out their cytoplasmic contents and push it into the developing oocyte as the oocyte begins to get bigger and mature. And part of this process involves border cell migration, as shown here as this tiny cluster of cells, which is actually initially born on the anterior end of the egg chamber and needs to migrate through and between the nurse cells to get to its final destination point, which is between the nurse cells and the developing oocyte. And the border cells are really important because if they fail to migrate, you don't get proper development of the oocyte. And their function is thought to aid in the formation of the micropyle, which is going to be used later for sperm entry into the oocyte, as well as providing a signal to allow for proper anterior to posterior polarity of this developing oocyte. So the way that the border cells move is actually as a collective cell migration, a cluster of cells that together move, again, between the nurse cells to its final destination point. And there's actually quite a bit known about how it gets there versus, up here or incorrectly positioned. And it's mostly through guidance factors that secrete out signals that then dock onto receptors that aid in migration, as well as local signaling that occurs between this cluster of cells. And what the paper will talk about on Friday is who's the leader? Who's the ringleader of this group of cells? Is it that everyone kind of works in concert? How do they communicate with one another? And this is different from the single cell migration that we've seen before because single cells are isolated, so they can make their own decisions. What happens when you're actually connected to a bunch of other cells? How do you work together for the group of you to migrate through, again, between these cells to get to your final destination point? And we'll talk more about that on Friday. But in order to understand the paper, there's one technique that they sort of use over and over again, and it's a technique to look at tissue-specific expression of a gene product, and it's the UAS-GAL4 system. So I'll just briefly talk about it here, and we'll talk more about it on Friday. You may have already learned about it in G3, and if you have, then this will just be a refresher for you. But the idea is that there is a sequence, which is the UAS, the upstream activating sequence, that allows for expression of this gene product. But UAS by itself is just not enough. This gene is in its off state. What it requires is a protein called GAL4, which then docks on to the UAS sequence and allows for proper gene expression to then get your gene product. The way that you get the tissue-specific expression of this gene product is you make GAL4 under the control of a tissue-specific promoter. So you have as a promoter, say, muscle cell promoter driving GAL4. Then only in muscle cells do you get GAL4 transcribed and translated. Then that GAL4 could dock on to the UAS site of this gene product and make gene product only in muscle cells. So the tissue-specific promoters that they use in this paper, this SLBO, which is specific for the border cells, such that you only drive gene product expression in the border cells. So what it looks like is you take a fly that has the border cell promoter driving GAL4, and you cross it to another fly, which has UAS gene product. And again, this by itself will give no gene product. However, when you cross them and you look at their progeny, and you have a copy of this transgene and a copy of this transgene, what you get is GAL4 docking onto UAS, and then you get transcription and translation of your gene product, again, specifically only where this promoter is active. In this case, only in the border cells. Okay, so this hopefully will help guide you through the paper and help you understand the experiments that they did, and we'll talk more about it on Friday.